Hello, everybody, and welcome to our CondaLab talk. We'll take a couple of minutes just to wait for remind attendance. Okay, thank you. More still people connecting, so. Okay, I think that we can start. Good morning, everyone. Thank you and welcome to our CondaLab talk. I'm going to introduce myself. I'm Jaime Martinez, Product Manager at CondaLab for microbiological products. Today, we are proud to launch our new solutions for the analysis of cosmetic products based on our CondaChrome technology. I want also to introduce to Amparo de Benito, Head for Microbiological Models at INEA, Technological Center located in Valencia. Good morning, Amparo. Yes, the microphone.
Hello, hello.
Hello. Amparo has a bachelor's degree in biological science, specializing in biochemistry and food microbiology. She has also a postgraduate diploma in food technology by IATA, the SIG, Technological Center here in Spain. As I said, she has developing since 2000 at INEA some projects and has some over 7 years of scientific experience developed at the IFE Food Research Center in the United Kingdom, where she was collaborating in predictive models and innovative inactivation treatment like high pressure. She was also part of INPIVA's high specialization program at Industry Technologies, which allowed her to spend 3 months stationed at the University of Connecticut in USA. Since 2016, she has been the national coordinator for the Committee for Standardization of Microbiological Methods in Food for UNE Group GT6 of CTN 34 plus CC4 and representative of the national group in the European Committee for Standardization and the International Organization for Standardization ISO. Also, Amparo has been the person who coordinated the validation and interlaboratory study for the product that we will present today. I take this opportunity to thank you for her full willingness to participate with us. We are very happy to have such an exceptional guest today with us. Well, one introductions were made. Let's see what we have prepared for you in the first part of the talk. I think that will take around 45 minutes at least. I will do a small retrospective of the microbiological analysis of cosmetic products to finish by presenting the new solutions that we have prepared for you. Once I finish, Amparo will take the floor to present the carried out studies, as well all the obtained conclusions that she got in this study. So I'm going to share my screen. Okay. Okay. Please, Amparo, can you confirm that the presentation is showing?
Yes, it's showing.
Yes, perfect. Thank you, Amparo. Okay, let's get started. And as I said, we are here. I'm going also to put off my screen. Let's go. Okay. As we said, we are here to present our CondaChrome technology, chromogenic culture media for microbiological analysis, and two products, CondaChrome PEC and CondaChrome Burkholderia. But before we went there, and as we said here in the index, we will first talk about the alternative methods present in the market and how CondaChrome can become our best ally. We will continue with a review about cosmetic product testing, what regulatory environment have these products, what microorganisms are analyzed, and what are its standardized and proposed methods in the regulation. And finally, and as a highlight, we will present our new solutions for the cosmetic industry. The solutions are alternative methods which have been developed by Conda Lab and supported by INEA in their validation. Okay. So, we are all aware about the time involved in a microbiological analysis by conventional methods. Due to this, more and more laboratories are implementing alternative methods in their practices. And why? Because there is a need of fast and available results as consumer health and company reputation come into play. Likewise, sensitivity, ability to detect microorganisms at low concentrations, and specificity, avoid false positive are also key points when we decide to implement this kind of method. As you already know, there is a great offer of quick methods in the market, from molecular diagnostics by qPCR for detection, pathogen detection, to immunological tests, for example, also the mass spectrometry such as Malditov, or new techniques like next generation sequencing, which is increasingly implemented in clinical diagnosis, and this coming to the safety food war at the moment. But not all are advantage. These techniques also show some disadvantages. First, not all laboratories are equipped to implement these ones. High initial investment is required, both to prepare the laboratory and to acquire different equipment or reagents. And of course, staff training is also to be considered. What do we want to offer you with CondaChrome? Okay, so we want to bring to you the solution with all advantage of alternative methods and techniques, but adapted to any kind of laboratory. And no investment is requiring a specific equipment, and the same staff who works daily in a microbiological laboratory will be fully capable to use our culture media. So, what are the advantages that it brings and make CondaChrome a highly effective alternative methods? In 24-48 hours, once the plate is inoculated, you will be able to see the results. Each colony, thanks to the CondaChrome technology, will be shown with its characteristic color, allowing its identification with the naked eye. We know that experience is a degree, and more when we talk about microbiology. It's not easy to identify the characteristic colonies of a certain microorganism. But with CondaChrome, it will be easy even for the most inexperienced technicians. On the other hand, in some of our CondaChrome culture media, such as the one we will present today, you will be able to see different bacterial groups in a single plate, saving time in the preparation of media and space in the number of plates. Furthermore, our CondaChrome culture media have a higher sensitivity and selectivity than traditional culture media. And maybe you may be wondering, but how can a sample color detection be so effective? Well, CondaChrome bases its technology in 3 points. First, CondaLab culture media. Second, chromogenic substrates. And lastly, bacterial enzymatic reactions. 60 years as manufacturers of culture media have given CondaLab experience in its manufactures and development, obtaining great knowledge about the nutritional and selective components in all kinds of media. Once we have developed this selective culture media, we add chromogenic substrates. These substrates are colorless compounds that, when they are hydrolyzed, release a chromophore to the media. This chromophore is internalized by the bacteria, and then the colony will be presented this characteristic color, unique one. And all of this could not happen without the specificity of the enzymatic reaction. These enzymes hydrolyze the compounds present in the medium to release the chromophore. The chromophore is internalized by bacteria, as I said, and this is the unique color compared to the rest of bacteria, due to this specific bacterial enzyme activity. So. Then, okay, so if this has, I think that this one. If this has not convinced you about the suitability of using CondaChrome culture media, let's look at different examples how with this sample mechanisms, sample in quotes, it gives a high specificity to our media. And the first one, for example, beta degalactosidase is a characteristic enzyme of the coliform group. The chromogenic substrate known as salmon gall is hydrolyzed by that enzyme, releasing the chromophore to the medium and giving this bacteria a pink reddish color exclusive to this bacterial group. It should not be forgotten that the composition of the media has been developed for the growth of these microorganisms, so it is already selective. Just we are increasing this selective characteristic. Another strategy, for example, is due to the included in CCA medium, for example, is the differential reaction against different chromogenic substrates. Escherichia coli, like a coliform, would hydrolyze the salmon gall. So how we can differentiate it from the rest with a second chromogenic substrate, exclude. This is hydrolyzed by beta-D-glucuronidase enzyme, unique and exclusive in E. coli, and not in the rest of the coliform. In this way, instead of giving a greenish blue color due to the presence of exclude or a pinkish one due to the presence of salmon gall, it will give rise to colonies with a characteristic dark blue and violet color that will differentiate E. coli from the rest of coliform. So this color comes from the hydrolysis of both substrates, salmon gall and exclude. And finally, we can also play with the inhibition of this enzymatic reaction. For example, in our CondaChrome Vibrio, we can identify the growth of Vibrio alginolipticus from the rest of the species. In this media, the beta-galactosidase activity is inhibited by the high concentration of sugar present in the media. giving rise to colorless colonies and differentiating them from the rest of Vibrio species, which grow with a greenish blue or pale pink color. So once we have an overview about the CondaChrome technology and what is basically the new product that we will launch today, we want to do a retrospective about cosmetic product analysis. So, although cosmetic products are protected in their formulation, they are potentially prone to microbial contamination. There may be a failure in the preservative and mistake made along the manufacturing process or all the failing are factors that can lead to contamination of our cosmetics. Like there are not aseptic products, the regulation requires a microbiological analysis whenever the cosmetic is considered as a risk cosmetic. So for that, we can divide the analysis into large groups. One of them is the analysis of the microbial loads present in the product. And the second one will be the investigation of specific microorganisms. The analysis of mesophilic microorganisms include the analysis of bacteria, yeast, and mold present in the product by isolation and enumeration. On the other hand, the pathogenic microorganisms of obligatory reports, Pseudomonas aeruginosa, Staphylococcus aureus, and Candida albicans, are analyzed together with hygienia indicator, Escherichia coli, which its presence reveals poor hygienic practice during the handling and production of the cosmetics. Also, we have commented that these are the analyses established by the European regulation. The manufacturer must ensure that the products reach the safe to the consumer. So the health of the consumer and the brands, the manufacturer brands, are also at stake if we do not ensure that we put safe products on the market. Other microorganisms not contemplated in the regulation may also be interesting to analyzing and monitoring, depending on the potential contamination on the process or their usage raw material. For example, this Bacillus, Clostridium, or Burkholderia, among others, will be interesting to be monitored. Then what are the accepted limits in cosmetic products? This slide is only for information purposes, since these criteria are well known to the industry. And obviously, we all are following them. According to the ISO 17516 standard, we must find 100 CFU in category 1 cosmetic products and less than 1,000 per gram or milliliter in category 2 products. The category 1 for cosmetic has the products for children to apply around the eyes or the mucous membrane. So, and the result for all specific microorganisms to accept batches as suitable must be in all of them absence of the microorganisms in 1 gram or milliliter. Okay, then in this slide, I'm going to summarize what will be the analysis that we must do for a cosmetic product sample that has reached to our laboratory, and what is the workflow we should apply. We start from a sample of cosmetic products for which we take 1 gram or milliliter of each and make it a 110 dilution with a neutralized shea broth. For example, the English broth or others that are described in the standard method. And the objective of the target, in addition to dilute in the sample, of course, is to block the presence of preservatives in our product. If these preservatives are not neutralized, we take a chance of getting a false negative, since microorganisms may be there, but the presence of the preservative is inhibited them. We let this broth with neutral ulcers about 20-30 minutes, and then we made our serial dilution. up to -3, -4, depending on the expected load of the bacteria. Then we will inoculate our dilution in TSA, and TSA for the bacteria, and ESD for this amount, for the numeration. This inoculation must be done in duplicates. Plates that after incubation show more than 300 colonies will be discarded. Once we have count the suitable plates, the results must be expressed in CPU per gram or milliliter. Sorry. In the same tube that we use to do our Australia's dilution, it's incubated for 24-48 hours until the growth of microorganisms becomes evident. Usually, this growth is visualized by the turbidity in the medium. Once we have confirmed a positive growth, we will perform analysis of specific microorganisms. This part here. In this case, it is not necessary to make dilutions because we need to analyze the present absence of microorganisms. We must make sure that if the bacteria are present, it will grow. To do this, we inoculate 1 milliliter of the broth in a selective agar by duplicate. We will need 1 selective agar for each microorganism, in total 4 media, which are cetrimide for P. aeruginosa, McConkey for E. coli, Bay Parker for Staphylococcus aureus, and SDC for Candida albicans. These plates are incubated for 48 hours. The usual situation in the industry is to find these results are negative and there's no growth. Due to the production process is purely monitored and validated. If we find growth, we should do its respective confirmatory test, which usually take between 2 and 4 days. The presence of this microorganisms rules our batch out. Well, when we have seen what the standard methods according to ISO, we are going to present our 2 latest developments for this end of the year, CondaChrome PEC and CondaChrome Burkholderia. These products have been developed by CondaLab and validated by INEA for 5 categories of cosmetic samples. As I mentioned at the beginning, I will make a presentation of both products and then the validated methods, so that later in the most interesting part of this CondaLab talk, Amparo will take about all the results and conclusions obtained in the different studies. So here we have, we present the first of the products, CondaChrome PEC. The name of the product is determined by its main characteristic, the simultaneous detections of the 3 of the 4 notifiable pathogens in cosmetic products. P from Pseudomonas aeruginosa, I from Escherichia coli, and C from Candida albicans. Is she right? Well, as I said, the main advantage that this method provided is the simultaneous detection of these pathogens. And what else this implies? Obviously, by using just a single culture media, we will reduce the number of reagents, going from using 4 media to 2 to perform the analysis of specific pathogens. And therefore, by reducing the number of media and its manipulation, we will also reduce the possible mistake that may appear. Also, as it is a chromogenic medium, we will obtain a better presumptive identification on the plates, allowing us to speed up decision making regarding actions to be taken. The method, as Amparo will later confirm, has been validated according to a standard 16140 for the validation of alternative methods. In it, the total equivalence between CondaChrome PEC method and the ISO one is shown here in this table. We have obtained this data for the different pathogens in their own efficacy, specificity, and sensitivity. On the left side of the slide, you can see a chart with the proposed method by CondaLab, comparing with the standard method one. We can see how, thanks to CondaChrome PEG, we will reduce time and number of uses. In this slide, you can see how the different bacteria may grow on a CondaChrome PEG plate. Pseudomonas aeruginosa grows in very beige yellow colonies like this one that I'm putting with the mouse. Escherichia coli will grow in pink colony like these ones, and Candida albicans shows blue-greenish colonies that this small one. In addition, not only by color we can identify the bacteria, but we have also included an additional confirmation by fluorescence. Under UV light, the Pseudomonas aeruginosa and E. coli will show a positive reaction, so it's fluorescence positive, while that Candida albicans will be negative. This way, we will obtain much more confidence in the interpretation of our results. And here is our last launched solution today. Is for an unknown pathogen, although it's becoming less. It's becoming less so. Burkholderia cepacia refers to a bacterial group previously considered as Pseudomonas that comprise about 70 species and that is increasingly present in the production of sterile products and cosmetic compounds, being considered an emerging pathogen for the industry. As it is considered an emerging pathogen, we currently only find one standardized method in the USP, the American Pharmacopoeia, and this method is tested for pharmaceutical samples, not for cosmetic products. In this absence of this, many of the laboratories will fine-tune their method based on this standard, but updating to it to the needs of cosmetic samples. Thanks to CondaChrome Burkholderia, we have developed and validated methods for the analysis of these species, specifically in cosmetic samples, and the method is sample. So from our sample enriched sample and block with neutralizing broths for 24-48 hours, we inoculate 1 milliliter of the broth on our CondaChrome. Oh, sorry, here there is a mistake because this is CondaChrome Burkholderia that we are talking about. So, as I said, we enrich the broth and then we will inoculate with 1 milliliter of the broth our CondaChrome Burkholderia plates. In 24 hours, we will check results to determine the presence or absence of this microorganism. As I said, the validation protocol based on ISO 16140 has been followed. Although in this case we don't have a standard method to compare, I think that later Amparo will explain deeply how the study has been carried out, but it was comparing with different methods implemented in laboratories, which participates in the interlaboratory study. We have obtained an equivalence of 99%, so that was a really good results. And here you have a picture showing how the reading is in this culture media, getting a brown pinkish colony, characteristics for Burkholderia cepacia species. Candida albicans can also appear on this media, but it grows transparently and with a moderate development. It's not usual to find them. So, as you have seen, these are our two launch solutions present today that presents a series of advantages that can make life in the laboratory easier for you. Reducing the number of used culture media to eliminate potential errors allows us to prioritize and improve technical work in the lab. Not only that, also its easy implementation will not take more time than necessary. As always in the laboratory and everything in life, time is money. So I hope that you find this Conda Lab talk interesting. I hope you have not gotten too tired and you are willing to listen to Amparo. Thank you all. Thank you, Amparo, and welcome. It's your time. I'm going to go to give you the control. Okay. Amparo, perfect. I'm going to go out my camera.
Okay. I hope that you can hear me perfectly.
Yes, we can hear you.
Okay, and you see the screen.
Yes.
Good. So thank you very much for Conda to invite me to this webinar where I'm going to present the validation results of the study that we did with the media that was developed by Conda, that is Conda, sorry. And first of all, I would like to give a few words about the place where I work. ISAINIA is a non-profit private association dedicated to research in many areas. Our priority lines are food and health, food quality and safety, design and industrial production, environmental studies, and consumer studies. We do our research through many different technologies, which are mainly food technology, biotechnology, nanotechnology, electronics and communications, chemical technologies, environmental and energetic technologies, and packaging technologies. And we work in many sectors. Originally, INEA started in the food sector only as a food research institute. However, we are already working for 30 years, and through all this time, we have been increasing our scope of action to other sectors such as energy, packaging, chemistry, cosmetics, and pharma. Just for the ones that you don't know where we are in Spain, in Valencia, and we have large facilities in the Technological Park of Valencia. We have 8 laboratories that we can mention, that, for example, the chemistry, pathology, microbiology, molecular biology, and it's important that we have a biosecurity laboratory, which is category 3, and is one of the few private security laboratories in Spain, which allow us to do experimental work with pathogens that they are important. And they are really dangerous to work in a normal laboratory. We also have 12 pilot plants, which give support to our research in all these technological areas that we work. And to mention a few, for example, we have artificial vision, we have supercritical fluids extractions, we have new development of products, microencapsulation, and lately we have gone to the roof of our building to build microalgae cultivation that you can see the picture here. This is actually in the roof of our institute. So we're growing and taking the space, all the space we can. Okay, so now to center in the actual project, I would like to present this. The objective was to do a validation based in the ISO 16140 part 2 which is a standard that is to compare alternative methods against reference methods for microbiology. The objective was to compare CondaChrome PEG that has been presented by Jaime and is able to simultaneously detect E. coli, Candida albicans, and Pseudomonas aeruginosa, and compare it against the ISO reference method for control of cosmetic products. And in addition, we also did a validation of the method CondaChrome for the detection of Burkholderia cepacia. And in this case, there is no a standard such as ISO for Burkholderia. So we compare it with a commercial method that is quite implanted. So just a few words to say why we use the ISO 16140. This was done because the idea is to certify the media, and for that, the protocol that is approved in Europe is the ISO 16140. This protocol was developed by the committee of ISO for food microbiological methods, but is the only protocol that we were able to work that is very extensive and thorough in order to be able to certify the culture media in Europe. And just to tell you that this protocol has existed for a long time and recently has been reviewed, and it consists at the moment in 6 parts. And that's why we're using just the part that is to do with the validation of alternative methods against reference methods. There are other parts, for example, for the verification of methods within a single laboratory, which is very important, for example, if you are accredited. And some other protocols are designed for other uses, but we actually use the number 2 that is fit for this purpose, because it allows to prove the possibility of certifying a method that is a commercial method. The purpose is to be able to be commercialized. This protocol consists in two phases. One is the actual comparison study of the alternative method against the reference method, using many different samples, and as we will see later. And this is done by an expert laboratory. And then the other part is an interlaboratory study that will produce the results that the actual method is possible to be used and is easy to apply for any laboratory. So it's compulsory in order to get the full certification of the media to have interlaboratory studies. In this presentation, I will show you just the results of the expert laboratory, because we still haven't finished all the interlaboratory studies we want to do. And just to show you what actually we're doing is to compare our ISO method against the alternative method. And when most of the points lie on this line, means that they are equivalent because they give the same result. This will be, for example, for a quantitative method, but it's a nice image to make you understand a little bit about this. We compare, as we said, the methods for ISO, and here are the references of the ISO. In the case of Burkholderia, we use a commercial method, which is using the media Bacillus cereus, Burkholderia cepacia selective agar. And for the rest, as you probably know, for the Pseudomonas, we use the cetrimide. For the Candida albicans, we use the saburochloramphenicol, and for the E. coli, we use maconcave. The validation performance was done using different products that we will explain later. And we actually did a preliminary study to be able to inoculate the products with the different strains that they were the target microorganism to be detected. And also we checked that we were doing a proper neutralization step, which is shown here. And then we did an enrichment step and an incubation following the ISO protocol, which is temperatures between 30 and 35 degrees C during 24 to 72 hours of incubation. And then we played out in the two different media. So we did a parallel study. And the aliquots came from the same sort of bag. So it was very nice comparison of the chromogenic agar produced by Conda and the ISO agar. The incubation time were the same for both agar, 30 to 35 degrees C for 24 to 48 hours. And then we confirmed the typical colonies by a very fast method that we have in INEA, and you may know that is MALDI-TOF, is a confirmation through spectrophotometry. These are all the different categories that we study. As you can see, it covers all the cosmetics that they are susceptible of having this analysis. And we were helped by experts in the ISO committee for cosmetics in order to categorize the actual products. And then we choose from each of these categories, we choose different products that they were representing different areas of use. For example, for the hand, the body, the hair. And as you can see in brackets here, we have a number, which is the number of samples, different samples that we analyze for the validation. So as you can see, each category, we analyze 60 samples divided in 3 different types of products, which makes a whole amount of 300 samples. So it was a very thorough and comprehensive study, which is following the ISO 16140. These samples have to be inoculated because quite obviously the majority of the samples of cosmetics in the market are free of these nasty bacteria, so we have to get strains. And for each category, we use a different strain of each of the types of microorganisms in the scope, which is Pseudomonas, E. coli, Candida, and also Burkholderia, which I haven't included here, but also was done for that. It's important to say that for each of these 60 samples, we use different levels of microorganisms, and some samples were free of these microorganisms, so we could have also negative results. So it's important that when you do a validation, you have positive samples and negative samples in order to assess the method for positive or negative deviations. Here you can see that most of the origin of the samples were clinical or environmental, and also we have one that was from human feces. Within the study, it's important to do also a check that the actual media is suitable for a large amount of microorganisms of that category, which means that the majority of the Pseudomonas aeruginosus, for example, that we tested 50, the majority produce a suitable characteristics in the culture media, which are, as Jaime has explained, each of the microorganisms produce different colors, and 2 of them, Pseudomonas and E. coli, produce also fluorescence. So it's very convenient because in case that you have any doubts, you can also put the plate under the UV light and check if the colony actually is producing fluorescence. So it's a double check for these 2 microorganisms. You can see that for inclusivity, we have very good numbers of recovery and producing characteristic colonies. We obtain at least 50 strains of each type of microorganisms from different origins. And also we check the exclusivity, which means that microorganisms that they are similar to aeruginosa, but they are not aeruginosa, like for example, other species of Pseudomonas were also producing negative results, which they are excluded from the media, which means that it's compulsory to do 30 strains of non-target microorganisms and check that they are actually excluded. As you can see, the numbers are very good, and exclusivity was fine for the 4 media that we tested. And the overall results of the validation are presented here, where we can see for each type of microorganism, the results of the 300 samples tested, and we can see that we have positive results for the reference method and the alternative method, and negative result for the reference method and the alternative method. And only in these areas you can see the deviation. In this case, the reference method will be negative and the positive method will be positive. And this will be the deviation. Only 2 cases in this case, and 9 in this one, in which was reference method positive or alternative method negative, were atypical results. We tested that all these deviations were within the acceptance of the method. So the media was properly validated and it met the requirements of the validation. So thank you very much for your attention, and I will be happy to hear any questions that you have.
Okay, Amparo, thank you so much for your presentation and all your work that you have done with CondaChrome products. So we will take, I'm happy because there is many questions, but regarding time question, we will only answer some of them. The remaining one, don't worry, will be answered by email. So all of them will be responded. Don't worry. How about that? So the first question, maybe it's not more interesting question in a technical point of view, but I think that is, I'm sure that is happy. CondaLab is very happy to read that. That is when will be available the simultaneous detection pathogens media. This media is CondaChrome PEC. I think that it's important to start sound the name of the product, and it's all of them, Burkholderia, CondaChrome Burkholderia, and also CondaChrome PEC are right now available. So if you need any kind of information, you can contact us across the micro@condalab.com or with your sales reps also. And we are very, very happy and excited with the media. We really believe that this media can solve some situations in cosmetic microbiological testing products. So the next question is asking about how they can, if they are interested in participating in the laboratory study, if they can do it and how can do it. That. So, Amparo, can you help us in this point?
Yes, you could write to us, for example, either to Jaime or to myself, telling us that you are interested in participating to the interlaboratory, and we will answer you, telling you when is possibly done this interlaboratory. It's important to say that we will send all the media necessary. You only have to check with your facilities that you are detecting the bacteria, but you don't have to use any of your own reagents. Everything is provided for you. We will also give you a report, so it will be nice to do this interlaboratory, which is totally free, of course. And you could see and compare your results with other people, and that could be also good for you and your quality check. So we
on this point, Amparo, it's interesting also to say that the interlaboratory studio has to be done at the same time. So we need time to receive all the participants in order to going on with the study, right?
Yes, yes. Thank you, Jaime. We have to organize simultaneous because in an interlaboratory you are comparing your results with many other laboratories, which means that they have to be done simultaneously and with the same type of samples. And for that, we will have to see, maybe it couldn't be done next month, but we will try to see when it will be possible. Once we get all the laboratories and agree on a specific time.
Thank you, Frank. Okay, there is other question that is asking about if this validation that we have done with INEA justify the implementation when they will receive maybe an audit if they are accredited organization.
Yes, I mean, the idea is just that. I mean, many of you probably are accredited by your own accreditation bodies, and as you know, it's not possible to introduce a new method that hasn't been validated or doesn't have like a certification. So this validation can be used to justify that you are using this media, which is it's been already validated against the reference method, because when you have a specification, you always have the reference method that you can use, but you're not obliged to use the reference method. You can use an alternative so long it's validated.
Perfect. I'm going to answer a few questions more. More questions. One of them is which neutralizer broth they can use with the methods. I understand they can use whatever broths included in the standard method ISO, the English modified leaching, maybe also with peptone water with neutralizers. All the media that are using the method can be also used with CondaChrome, right?
Yes, I mean, as we did the validation, here we are talking about a culture media, which is the agar. So it's nice because you don't need any equipment special for this. And you use the same method as you has always used, but instead of 4 plates, you use one. Well, instead of 3 plates, you use 1 plate, which is very nice. And also the fact that you have a possibility to see different colors and to see ultraviolet fluorescence means that it's easier to interpret as well. So
a double confirmation. Okay, And the other one is more like a practical question. Don't worry about the presentation and all the information, because tomorrow will be sent an email including the recorded version, your certificate of participation of this Conda Lab talk. And of course, we are open to answer and receive any kind of question. We keep this one that are not answered for today, but along this week will be all the questions answered by email. So we are going to close our Conda Lab talk. I want to also say thank you to everybody to attenders. I hope that you find interesting the speech. And also, Amparo, thank you so much for your attendance and all your knowledge about the microbiological methods and their validation. And we hope to see you soon in other Conda Lab talk. Thank you so much, Amparo. Thank you, everybody. Have a nice day. Bye-bye.